Hi y'all, this is Mr. Labonte. I'm going to be talking about the specific stages of glycolysis. That is exactly how a sugar molecule gets torn apart and turned into ATP. Remember, that's like the $5 bill of energy. And as we look at this, I'm going to use this video and pause it to explain some components of this video.
Compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules
All right, so there was a fair amount of stuff that was covered there. I'm just going to review a few things before we start. So this is over here, this is the cytoplasm. Right, so we're out in the cytoplasm. This is the mitochondria that's being shown kind of here in the back. This is a carbon molecule, a sugar molecule, and you have carbons here, 1, 2, 3, 4, 5, 6. So this is like a glucose molecule. That would be the oxygen there, so that's not too surprising. And you may remember from class that ATP, adenine triphosphate, well, I won't write the whole thing, but just say this is TRIPHOS. So that means there's 3 phosphates here, and what you're just about to see is ATP is going to turn into ADP, and that is it's going to lose one of the phosphates, and the phosphates are going to connect to the sugar molecule. And so when the energy investment phase occurs, ADP has lower energy than ATP, so we're actually giving energy to this glucose molecule, which is the molecule we want to get energy from. But we have to give a little energy in order to start this to work. It's kind of like lighting a candle or something like that. And if you think about it, that's a good idea, because you don't want sugar spontaneously releasing energy in your body. You want to have to give sugar a little bit of energy in order to get the energy out. Otherwise, you would, all the sugar would just blow up in your body and you would spontaneously combust, which is not something you want. So I'm going to try to make this a little easier to erase. Not having a lot of success with that. There. Let's see if this works better. Okay. So after I erase this, we'll get back to the video.
Transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two...
All right, so what you just saw was a molecule NAD+, and that came by and it got turned into NADH. What that means is you had one electron that went to this molecule and that took care of the plus, and you had another electron that was bonded with an H, right? So you have a hydrogen atom and an electron. Those two things join to make this molecule. And what is very important to understand is this molecule is now high energy. So the sugar has just given some energy away in the form of this NADH, and we're going to see this in the electron transport chain. So NAD plus is low energy, NADH is high energy.
Two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced.
So this is just going over the first part of respiration, which is glycolysis. And again, to reiterate what they said, you got 2 molecules of ATP, 2 high-energy NADH, and you still have these 6 carbons, so there's 2 3-carbon sugars that are floating around. So you still have a fair amount of energy that's yet to be extracted from this.
When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the
All right, so this step is super complex, and you don't really need to know anything about this step. But I do want to point out, well, you need to know a few things, but really not a whole lot. What I want you to understand is you have 2 carbons that start the cycle, and so that makes actually the 6-carbon molecule because you get 2 carbons are added to the existing 4. And then as that happens, we're extracting the hydrogens and the electron that goes with the hydrogens in these NADH molecules. That's going to leave these carbons from this molecule with less and less energy, and eventually they are going to leave along with the oxygens. Remember, C6H12O6 has oxygens, so those oxygens are going to leave. So from the original sugar molecule, you're losing carbons and oxygens here. The hydrogens are going somewhere else to the electron transport chain. Again, you see more hydrogens and more electrons are leaving. And then the other thing to note is that water is being added. So in the original equation, I showed you that water was part of the first part of the equation. This is where the water is going, and that's where you're getting these extra oxygens from, so that you can keep producing these carbon dioxides. You are getting some ATP out of it, and you're getting FADH2, which again is really just a mechanism of taking the electrons and the hydrogens and bringing the electrons and the hydrogens somewhere else. So this is what you're getting out of it, a little ATP and a lot of high energy hydrogens and electrons.
Majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another.
All right, so the first thing to get here is basically the architecture of this. This is the outside of the mitochondria, and this is a double lipid bilayer. And then this is the inside of the mitochondria, and this also is a double lipid bilayer. Hopefully you can see these are phospholipids, right? And so what's really important to understand is that area right there is hydrophobic, which means it does not like water and it does not like charged particles. And I'm going to write no charged particles here. So it does not like charged particles. So that's essential. And then understand that you have these proteins, and these proteins cross the membrane. So because they cross the membrane, charged particles can go through these proteins from one side to the other. But if you have a charged particle on one side or the other side of this membrane, it's not going to be able to get through. So that's basically like a dam. All right, then the second thing you're going to see here is our friend NADH. So this is that molecule, and remember, it had hydrogens, which had high energy, and it had electrons, which are high energy, and it's going to drop these electrons off, and the hydrogens, which in this case are going to be represented by a little plus sign because they really are just a single protein, are going to be let go too. And then this molecule is going to be NAD+, and it's going to go back and get more hydrogen. So you can think of this as kind of like a little dump truck. It goes and picks up hydrogens and electrons and then brings them back and dumps them off. The same thing is going to be true. Remember, there's this other version of it, of FADH. It's going to do the exact same thing. It's going to drop off electrons and it's going to drop off the hydrogens and then go back and do something else.
As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane.
Okay, I want to review what you just saw, which is you saw these little purple things, right? And these are the hydrogens, and they're called hydrogen ions because they're missing their electrons. You saw them shoot through really, really quickly, right? So these purple things are going through. These blue things are the electrons. These are the electrons that would normally be connected to the hydrogen, but they're not there anymore. So I'll erase that little line. They're not connected to the hydrogen anymore, and they're traveling along the membrane. So they're trucking along this way, and as they truck along, what they do is they allow these proteins to grab hydrogen ions and pull them through. So what you're getting is you're getting hydrogen ions getting pushed from this side of the membrane over to this side of the membrane. More and more and more hydrogen ions on this side of the membrane. Remember, this is a phospholipid bilayer, so even though diffusion would say that all particles want to be equal, these hydrogen ions cannot go through the other side because that's hydrophobic. So they get trapped on this one side.
When the hydrogen ions flow back across the membrane through an...
So she just talked about the hydrogen ions flowing back across the membrane. Here, I can make it purple. I'll make it blue. So these hydrogen ions, because they're all over on this side, and there's a bunch of them over here, and there's not very many here, they want to go back across. They go back across through this very special molecule called ATP synthase. So you should remember that synthase is like to make. And so this molecule uses the energy of these hydrogen ions going across to take ADP and turn it into ATP. Now, why is it that these molecules have energy? They have energy just simply because there's a ton of them over here, or these ions, so these are little hydrogen ions. There's a bunch of them over here, and there's not very many here. So you can think of it as like a dam where you have a bunch of water on one side of the dam and not a lot on the other, or whatever you want, but it wants to go through, and because it's polar, it can't go through the phospholipid bilayer, so it can only go through this molecule. And that's where we get the energy turned into ATP, which you guys will remember is the $5 bill. Okay, now the next question is, well, if you have all these hydrogens going over here, right, don't you just ruin the concentration gradient and then eventually this thing won't work?
ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain.
This is Mr. Labonte.
All the high-energy...
All right, so let's take a careful look at what was just said. Hopefully you will recall that the electrons were dropped off either here or here if it's FADH2, and they traveled across the membrane. As they travel across these proteins, they do work, and that work is in the form of taking hydrogens, or helping the protein take hydrogens and pump them out. So hydrogens are pumped out here, the electron does work, and if something does work, it loses energy. And then hydrogens are pumped across here. So these electrons, when they get to the end of the electron chain, are low energy. Well, there's only one molecule that's going to want something that's low energy, and that's oxygen, right? Because oxygen has a very high electronegativity. So what you can do is you can split oxygen apart. So remember, if you split oxygen apart, it'll have 1, 2, 3, 4, 5, 6 electrons, right? So it's going to want another electron, and it's going to want a hydrogen, a proton. So this electron and a proton is going to connect. Another hydrogen will connect with another electron that comes out, and you'll have oxygen with its original 1, 2, 3, 4, 5, 6 electrons. Then it will get two, here, let me erase a little bit of this just so it's not too ugly here. Then that oxygen will get 2 electrons, now they're in red, sorry, but from the end of the electron transport chain, and this wouldn't be happening all the way down here, but I'm drawing it out here just so you can see it. So that's what the 2 red electrons are, and I'm going to erase those lines. Okay, so those are our 2 electrons, and then I guess I'll use, I don't know, blue, and then the hydrogens that are coming through here, they're going to bond here because the hydrogens are going to want an electron, right? Because these are H+ ions. And so then you're going to form a new molecule, H2O, right there. And that is, when they say the terminal electron acceptor, that oxygen is the terminal, meaning the last acceptor of electrons. And so I hope that you can see, if you don't have oxygen, these electrons have nowhere to go, because nobody loves a stinky old electron that's all used up, right? Oxygen's the only thing that'll take it. So this whole system is going to back up. Well, if this whole system backs up, there's no need for NADH or FADH2, so these back up. So if all of this stops, the only thing you have is that thing right at the very beginning of this story where water was split, where glucose was split at the beginning. Without oxygen, the entire mitochondria shuts down, and that's why you get sore muscles generally. You get the buildup of lactic acid. We can talk a little bit more about that. Okay, so I hope that helps. I'll play the rest of this and see what it has to say as I erase.
Electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.